So this is a trihybrid cross. How large a Punnett square would you need to draw to predict the O-spin of the cell? Again, it's a self-pollination. So it's a heterozygous combination, meaning we can write it capital Y, simple Y, capital R, simple R, capital I, simple I. And when it's self-pollinate, again it's the same capital Y, simple Y, capital R, simple R, capital I, simple I. So list all gametes that could be made by the female. So gametes have half a number of alleles, and here there are 6. 1, 2, 3, 4, 5, 6. Gamete should have then 3. 6 become 3. Right. So let's try to first find the gametes. So this is a one cell with full number of chromosomes, and that cell is going to make the gametes. During gamete formation, we can use what we have learned so far, Mendel first law and Mendel second law. Each allele going to the gamete when the gametes are formed, and that is a randomly assorted into the gamete. So then we can write all the possible combination of these letters. What could be the possible combination of letters? So if we take capital Y, so this can combine with this capital R and then capital T. So sorry, not T I, right? Capital R, capital Y, capital R, capital I. And then capital Y with capital R, and then simple I. Then again capital Y, simple R, and simple Y. Then capital Y, simple R, and capital I. Capital Y, simple R, capital I. So then we can start with simple Y. R, I, and simple Y, capital R, I, simple Y, R, capital I. Then the last simple Y, simple Ri. So it is trying to combine 3 letters with all the possibilities, right? Because this one is a one homologous chromosome, this is a one homologous chromosome, and this is another homologous chromosome. So when we separate each chromosomes, that now in this case allele has equal chance to combine with the other chromosomes. So that's why we had the first capital Y, capital Ri, and the last simple Y, simple Ri. So in between, we can find all the possible combinations. And as I changed this letter here, and I changed this 2 here, and then like this, I change this and this here. So there are 8 gametes. This side has 8 gamete, and this side 8 gamete. There should be 64 then. 64 squares in this trihybrid cross in the Punnett square. Is this clear?
Clear, sir.
Okay, then what I do, I will send you again today another tute. This has old syllabus questions, and I included in these notes 2 questions from the new syllabus. That means 20, I think 20 and 21, no, like 21 and 20. So we should always get idea what kind of a question is asking. When we are learning whatever unit, we should add past paper questions to get an idea what kind of a question asking in the section. It is not like checking the knowledge. So any unit, if we include 1 or 2 questions to get an idea what kind of a question is asking. Now, genetic is different because genetic is application type. But in other sections, they are different. They are more theoretical questions, but here more application type of questions. So I think we should understand there is a less chance of asking the theoretical question from genetics. If you look past old syllabus, I think very hardly they have asked theoretical questions because they have to make a good application question in the paper. But in the new syllabus, a little bit changed the trend. You will see the difference doing old syllabus and new syllabus questions. New syllabus, difficulty level is less now than the old syllabus. Because again, I feel that they are going for more half application and half theoretical like that, like not completely application. But we should always expect application question, because theoretical question is not that difficult to answer. If you remember it, then you can answer it. But in case we never know these trends and the exam type questions, there's no limit. We have to prepare for any kind of situation. So they can't ask more than that, because this is the limit. So we have the limit. We learn everything within the limit. But when they ask application question, only they can change a little bit of the format, but the method they can't change. For example, I remember the notes section I sent you. Let's do that one. New share. Okay, where is the note? This one. Okay. All right. So the last two things to finish the Mendel genetic. We did this test. Here is the notes which I sent it to you. Yeah, that's the end. So dihybrid test cross. This is from again a resource book. Only the format is different, and the monohybrid cross. And today I did this application section, probabilities and prediction of inheritance pattern in multifactorial crosses. And I included here two latest questions from the new syllabus exam. These two questions. So this question is more theoretical type. There's nothing much about application. So let's try to do this question. Certain plants of a particular species bear purple flowers, while other plants of the same species bear white flowers. So that is something what we learn. The pea plant, purple flower and white flowers are there. To explain the inheritance of the flower color of this plant species, monohybrid cross is sufficient, dihybrid cross is sufficient, monohybrid and dihybrid cross both need knowledge of, okay, we haven't done this one, knowledge of, we haven't done this one, okay. Fourth and fifth one we haven't covered because it is not covered under Mendel genetics. Then we are learning Mendel genetics. So we haven't seen something under Mendel genetic like that kind of a thing. That means Mendel discovered the pattern of inheritance within his genetic. So anyway, fourth and fifth, we don't want because they are not within the Mendel genetic, because we're going to learn them as something like non-Mendel genetics. So which one you think sufficient?
First one?
Yes. If actually we learn straightforward that one, because Mendel did monohybrid cross, and after that he proposed his most of the 1 and 2, because F1 and F2 generations he just proposed, and then he went on to do the dihybrid cross, but monohybrid cross enough to formulate first law and second law. After that he went to discover more and more things with dihybrid cross. So monohybrid cross is sufficient. So this question is completely theoretical. There's no application. So then we should a little bit aware. They prefer to have mix of theoretical and application, but we never know it could be completely a application calculation type of question can be asked. Now the second question, which of the following statement, I think this is 2021, this question, right? Which of the following statement regarding the cross capital R, simple R, and capital R, simple R is correct? The probability of having the allele simple R in both the egg and sperm at fertilization is half. The fertilization is a formation of the zygote. That means if it is capital R simple R and if it is capital R simple R, uh, they form the gametes, and the gametes are capital R simple R, capital R simple R. And these gametes can fuse to make the zygote. So the probability is, I think we have learned many times, simple R is coming only one time, so 1/4, right?
So even capital R, simple R can no issue, is it?
Capital R.
Simple R.
No, I just address is having the allele R in both the egg and sperm. So both should have this one. So that means again, it is half in this case and half in this case. So then multiplication rule, it is 1/4. So having allele R in both egg and sperm. That means it's similar to the what we discussed about the dice. We just throw 2 dice and getting 1 and getting 1 out of 36. So having a simple R and also having simple R is half and half, it's 1/4.
Yes.
It's just a multiplication rule. And I think next one is pretty obvious. It's not a dihybrid, right? It's a monohybrid. This is monohybrid cross, not dihybrid cross. According to Mendelian inheritance, the probability of having dominant phenotype in F1 generation by interbreeding of F1 is 9 out of 16.
False.
Now this case, if we cross this one, so we get a capital R, capital R, capital R, simple R, capital R, simple R, simple R, simple R. Then this is the F1 generation. Now they say F1 generation is interbreeding now. So if we are trying to go for the F2 generation, we don't know which one we're going to use. For example, we can use a capital R, capital R. We have a capital R, simple R, and simple R, simple R. then the probability of having dominant phenotype. And they have given here 916 is the dihybrid cross 9:3:1 ratio. And we also look all the combinations, like we can have a capital R, like this. We can have this cross, and we can have this cross, and we can have this cross. Because F1 generation, these are all the possibilities. And F2 generation, we use F1. And it is not mentioned which one we have to use, which genotype. So these are the all possibilities. But today we have learned, if we use this kind of all combinations, we always get these ratios, these three ratios. We don't get 9 out of 16, which is dihybrid cross. So always monohybrid cross, we get these three ratios.
So capital R can also this thing with capital R, simple R, no? Sir?
Capital R with capital R, capital R.
Capital R, simple simple R, simple R. No, capital R, simple R.
Capital R, simple R, capital R, simple R is here.
No, sir. Capital R with capital R, simple R.
Capital R with capital simple R, simple R. Capital R with capital R, simple R. This,
Yeah.
yes. Yes. But again, if we look the ratios, R simple R, simple R, simple R. No, capital R, simple R, right? Again. Capital R, capital R, capital R. Okay. Again, one, no? Because all dominant. All are dominant.
Yes.
All are dominant. So mostly half, quad, and 1 are the ratios we are getting for monohybrid cross. So this ratio indicated is for the dihybrid cross. Okay, now fourth one and fifth one, we haven't learned.
So, can you explain how did you get the other part in the third question? That all are one. That
Can you tell which one?
that capital R, that Punnett square you drew?
This one?
No, sir, the second one.
This one?
Yeah.
Okay, then this is the capital R, capital R, capital R, simple R. This cross, right?
Yeah.
So then alleles, gametes are these, and here gametes are these two. Capital R, other one capital R, simple R. Then this capital R can combine with this capital R to make R, R. Then these two, capital R, capital R. Then this one, capital R, it should be simple R, right? This should be capital R, simple R. Okay, then the ratios are half and half again, right? Not the one. Half, half. These two and these two are the same. Then the next, if you take fifth one, when they use the word linked, the linked is something like this. So far, Mendel genetic, what we learned, this is a chromosome, and this is a homologous chromosome, and the allele located here, and the other one located other chromosome. But there is a situation we're going to learn. If there is a allele and there is another allele, the chromosomes, this allele located here and the other one is also located here. This is linked. They are the same chromosome. If they are the same chromosome, when gamete formation, they are not separating. So this is going to one gamete. This is against Mendel rule, because Mendel said when the gamete form, allele get into a gametes. And it's a very random process. But when it's linked, both are going to the same place. And imagine that we are talking about the purple color, round shape, like if one is located like this is a color, responsible for color, this is responsible for the seed shape. B is responsible for the seed shape. So they are not separating now. They are going to the same gametes. We never get the ratios like what we get from Mendel genetic. So that's what they say in linked gene. So if this is the way they given in the question, they are not linked. Because linked one we can't write like this. So this is wrong, this is wrong, this is wrong, this is wrong. And we haven't learned codominance. We're going to learn in the next section codominant. And in Mendel genetics, the ratio we are expecting is a phenotypic ratio 3 to 1. Because look at this Punnett square. Capital R, capital R, capital R. 3 dominant, only 1 recessive. This is the ratio, phenotypic ratio we should get according to this question. 3 to 1. And F2 generation, if we go F2 generation, in any of these cross, we get 1 to 1 here, for this case 1 to 1, 3 to 1, and all. So we are getting the ratios. You can draw Punnett square and check. We get this kind of a ratio. But you can notice here, there are 3 phenotypes. Because we always have 2 phenotypes, it should be a purple or white. But they say there is another phenotype, because 1 to 1, not 2 phenotypes. There are 3 phenotypes here. In the F2 generation. So we learn codominant in the next sections. When we learn codominant, it should be a answer should be then 4th one, because we need the knowledge of codominance to answer the question, and also we need knowledge of link gene to answer the questions. So this is not completely Mendel. It is also mixed with the non-Mendel genetic, which we're going to learn. But this one is quite easy. This one we can't still completely work out because we haven't learned other things.
So, for the third question, the probability 12 out of 16 is possible, na? The third point.
This choice, third choice.
Yeah.
Probabilities.
12 out of 16 is possible, na?
12 out of 16, like this?
Yeah.
Let's see. 3, 4. Yeah. 3 to 1. Yeah. This is 1/3 now. Yes. Yes, that is possible. 1/3. 4/3, right? 4/3. This is a 4/3, and this is according to a 4/3, and like this. So we get these ratios, and if we want to see in 16 times, 12, yes, this is possible. Yeah, that's a good point, I think. So if it is coming like a 12 out of 16, that could be tricky again. We had to figure it out. It's 75%. Yes, 75% is possible. This question, for any gene with dominant allele A and recessive allele simple A, what proportion of the offspring from capital A, capital A, capital A, simple A cross are expected to homozygous dominant? homozygous recessive and heterozygous. So capital A, capital A, capital A, simple A. Capital A, capital A, capital A, simple A. So homozygous dominant is homozygous dominant, these two. Heterozygous, these two. And we don't get homozygous recessive. So it is homozygous dominant is half, heterozygous is again half, and this is 0. Okay, this is, I think, a good question that you can practice. If they have these genotypes and without drawing a Punnett square, so you have to list all the genotypes and you have to calculate the probabilities. So without drawing the Punnett square, you can do this question. As we have learned today the probability rules, did you do this question? Yes or no? Have you done
No,
it?
sir.
Yeah, I think keep this as a practice question because we have learned today these probability rules. So you can practice and we can see a solution next time. This question I leave then because you learned today, you can do it now, right? Fourth question, we have answer. Again, flower color, seed color, and pot shape. 2 pea plants. What fraction of offspring are predicted to homozygous recessive for at least 2 or 3 characters? I think this is the answer for that question. This one. Simple P, capital P, capital P. Yeah. Similar to this, right? That question. You can also practice this question. This is also a good question to practice. Third and fourth one. So I leave these 2 questions, third and fourth. We see the answer next week, right?
Okay, sir.
Okay, there are a few questions. You can download the next topics. I will also send you the past paper questions, all syllabus. We can also look these questions while we are learning